Okay, great. So thank you for your introduction and hello everyone. Welcome to the second part of Biorelate webinar for knowledge graphs for drug discovery. As Mark already introduced, my name is Özge and I'm postdoc and also knowledge graph engineer in Boehringer Ingelheim. And today I will talk to you about knowledge graphs for indication expansion, how we implement it. So my outline consists of 3 parts. The first part is the fun part. I thought that maybe there are some audiences that has been heard about like knowledge graphs, but they just want to know about how they can use it, how they can implement it. That's why I just put this first part, Introduction to Knowledge Graphs, as 101. And in the second part, I will talk about how we implemented knowledge graph for indication expansion. And as bonus, I will briefly describe or discuss how we can use knowledge graph for knowledge exploration within the organization. So first, let's start with knowledge graph. What's a knowledge graph? So assume that, first I will start with a network, define a network. Assume that we have a protein interaction data in a tabular format, which defines this node 1 column defines the protein, and the node 2 columns define the protein, and other proteins and their interactions are defined in this relation column. If we want to convert this tabular format into network format, then we will turn the nodes, the proteins into nodes, bubbles, circles, and the edges, relations to edges, then we would have a network, right? But this would be a homogeneous network because there is only one type of node, which is protein in this network. If you would like to add disease information that are related to the protein, as relation, I mean just if they're published together, then this will be heterogeneous network because now there's more than one type of nodes, which are protein and disease. So we can define that knowledge graphs are actually heterogeneous networks. But what makes them different than networks is that because they have a semantic schema defined on top of them. So in this case, we have the semantic schema as that we have defined the entities and their relationships that exist in our knowledge graph. In this example, we have the protein class and we have the disease class. We also define that protein is interacting with a protein class and this protein has a relation to a disease class. And once we define this domain entity, the semantic schema, then we populate our ontology with the data in the instances, which then we have the knowledge graph in the end. So we'd say that this semantic schema is also called as ontology. So ontologies are formal conceptualization of entities and their relationships that belongs to a domain. So the term ontology comes from actually philosophy. It's the science of being, science of existence. So when we define a domain ontology, then we think about which entities that we should include and which entities we should not include, and the relationship between these entities. For example, if you want to make a disease ontology, then we would have the disease entity and the virus or bacteria that causes the disease, and the animal, the tissue, the vaccine, and the drug entities to make the domain ontology for the disease domain. This is not a complete, but just to give an idea to you. This will be our entities, and if you want to populate this ontology to build a knowledge graph, for example, COVID-19 will be the instance for the disease entity, and AstraZeneca, BioNTech, and Moderna will be the instances for the vaccine entity. And this is how we would have the knowledge graph. So maybe you would ask right now, what is the benefit of using this knowledge graph ontologies, and why don't we just use the networks? So here are some facts. The first one is that ontologies enable machine interpretability for the knowledge graph. From the screen, we just see these colorful bubbles that are connected to each other. But on the background, actually, they are defined with their resource description framework, which is a special syntax, and they explicitly define the classes, the entities, and the relationship between these entities very discretely and like a coding, so that the machine can interpret. And also, we also define the instances and the data inside the knowledge graph. And with this framework, the data, the knowledge, can be interpreted. So this is important because then this provides the semantics for knowledge graph. So what I mean by semantics is, I will give an example by corona. So the word corona, what comes to your mind when I tell you corona? Okay, right now maybe we are a bit biased, so probably most of you will think about the virus corona. But maybe some of you will think about the beer corona, or if you're Italian or Spanish speaker, maybe you will think about the crown as a corona meaning. But if I tell you that it's been so long that I haven't drank corona, then you will immediately understand that I am talking about the beer. How? Because you're human and you're intelligent, and from the context of what I say, you can understand what I'm talking about. But the question here is, how does the machine understand it? So I wrote a theory that I wanted to drink corona and to show me the pictures of the corona. And it bring me the right pictures of the corona as a beer corona. How it works is because there is an active ontology and knowledge graph in the background, which can disambiguate what I'm talking about from the context of my question and brings me the right answers. And thirdly, it enables reasoning and inferencing of the knowledge graph. So to explain what's reasoning and inferencing, I don't know if this will be a good example, but it mostly, I think it explains very well what's reasoning and inferencing. So this screenshot is from a TV series called Friends. I'm sure most of you heard of it or already watched it, and maybe you're already a fan of Phoebe, which I am. And she says in this text that she had a hard life and her mother was killed by a drug dealer. And her friend Monica says that your mother killed herself. And she says, yes, she was a drug dealer. So this is a way of her thinking, the way of she reasons, the knowledge she has. So we want this knowledge to be, so we are a human, we can make this reasoning by ourselves because we're intelligent, but we want this reasoning to be and inferencing to be done by the machine, by the knowledge graphs. So if you just look into this example very simply, we have the entity as a person and job title, right? So person has a job title and a person kills a person, let's say. And if we just define this inferencing rule that if a person is killed by a person who has the job title X, then that person is killed by the job title X, right? Then with this, by giving this information to the knowledge graph, it creates this link automatically that person is killed by this job title. So like in Phoebe's case, how she thinks is that her mom kills herself and she's a drug dealer. So with this information that we give the knowledge graph, the logic, the rule that we give, that her mom is killed by a drug dealer, so she's not incorrect, she's right. So this is why it's actually really nice to have the knowledge graph to define these logic rules for creating the links between the entities that doesn't exist in our initial data. And lastly, so knowledge graph supports question answering. Again, I found this picture in the internet where again they asked Siri about the mom's blood pressure. So it understands what the blood pressure is, and it also has a reference data that it can compare and give an answer to me that if the blood pressure is normal or high or optimal. So this is the sum of, I hope it was a nice summary of what the knowledge graph and how it can be fun to use inside our knowledge graph and how we can use it in our cases. So in the second part now, I will talk about how we implemented this knowledge graph for indication expansion. So our goal was to find new indications for molecular entities. So we had the target, which already had a validated pathway to a disease, and we were looking for alternative diseases, a novel pathway. But the question here was that what if this link between the target and alternative diseases were indirect? Let's say that the target was interacting with a protein, and that protein was interacting with another protein, and that was linking us to alternative disease, but that was not published yet, and we just wanted to connect the dots and find this unlinked information by ourselves. So if you need an example for why would that be necessary, here is one. So Propranolol preventing Hemangioma, it was published in 2008. So we had the knowledge that VGF pathway to RAF1 and cell survival was published in 1997 and 1998. Then in 2003, they published that propranolol was blocking VGF, and this was causing apoptosis, and it was published in 2004. And with this apoptosis, it was inducing hemangioma. So if we had just linked all the little information from extracted from papers or other data sources, we could see that there was a link between propranolol and hemangioma, and the latest date was 2004, but the link between propranolol and hemangioma was published in 2008. So this was one example. So with this example, we thought, okay, let's link all our data then. And then we also thought about the analogy from Google Maps. So in this example, I use Google Maps as my starting point. I entered Bibera, where we are located, to Istanbul for my endpoint. And Google Map extracted me this path between Bibera and Istanbul by using 6 different data sources, extracted the timelines from transportation web sources, extracted the timelines and integrated and linked all these timelines to each other to present me a shortest path between my start and endpoint. Sorry. And so with this analogy, we said, okay, let's conduct an exploratory study to see how we can implement knowledge graphs for indication expansion. So with as an ontology, we had this, we thought about using the disease, protein, gene, and tissues, and their relationships were defined so that gene had associated protein, and we had protein interactions, and this protein and genes were related to linked to disease, and gene had the expression data in the tissue and the cell type. And we had this protein interaction data, gene disease data, and gene tissue expression data. And so we just we built this ontology and we populated this ontology via using these databases. So we integrated all these sources in one knowledge graph. So we extracted the protein information, and we use this unique URI IDs for the entities or resources. For example, in this case, we use ensemble ID. Let's say we extracted VGF interaction to protein Y from one data source, and protein X interaction to VGF from another data source. But since they were defined with the same URI, unique ID, they were immediately merged in the knowledge graph, so it helped for the data integration. And then we traverse up to third degree neighbor of the given target. I will give an example in the following slides. This we just followed protein interaction change to the disease. And then we filtered the pathways based on their publication numbers and their tissue expressions. So that we try to filter out the proteins and diseases based on this if they have the same tissue expressions. And we also scored the papers, the links based on their publication numbers. And at the end, then we benefit from the reasoning and inferences to find this indirect link between the target and the disease. So how the inferencing was working, it's better with picture here. Let's say we had protein 1 interacting protein 2, and it was related to disease 1. And this protein 1 and protein 2 were expressed in the same tissue. This meant that protein 1 was one hop related to disease 1. And secondly, then we had protein 1 interacting protein 2. It was interacting protein 3, and it had related disease 2. And these 3 proteins were expressed in the same tissue. Then this 2 hop related link was created automatically, as same as the first slide. And how we benefit from reasoning and inferencing in this case is that, for example, some of the data sources was keeping the genes with entrance IDs, and some of them was using the ensemble IDs. But they were the same gene, but they just were expressed with different IDs, right? And then we just use all same as relationship to define that these 2 nodes are actually the same nodes. And when the reasoning was on, they automatically then exchange their relations with each other. So that, let's say, this ensemble gene was expressed in adipose, and when the reasoning was on, then this link 7124 was also automatically connected to adipose. And then we defined some inferencing logic rules, like if a protein was associated to a gene, and if this gene was linked expressed in the tissue, then it meant that we created automatically that the protein was expressed in this tissue, because usually the data we extracted, there was the gene and tissue, not the protein tissue. We just passed on the information. And another logic rule, we define that if protein 1 is interacting with protein 2, and if this protein 1 has expression in this tissue, and the second protein also has the same expression in the same tissue, and this protein 2 is related to a disease, then we created this one-hop related link between TNF-alpha and Crohn's disease in this case, automatically as one-hop related. So then we just conducted some example case studies to see how it works. So we looked for the links between TNF-alpha and Crohn's disease. And these were the top 3 ranked links between TNF-alpha and Crohn's disease based on the publication numbers, which are shown in the parentheses. And all these proteins were expressed in the same tissue. And then we checked in the literature and we saw that the role of TNF-alpha in Crohn's disease was with IL-8 and IL-6, and it was cited in 2011. And in another case, then we looked for C-ABL and Parkinson disease. So there was only 5 paths between this disease and the target. And we eliminated this ESR1 and PCNA because it didn't have the same tissue expression as these paths. And then from the remaining, we found that the link between C-ABL and Parkinson disease was via C-Jun. So this was just example cases to just give you an idea. So our experience with knowledge graphs are so far that it has some limitations because it requires time, effort, and people. And also the data quality is really important because there's so many noisy data that we want to avoid to have in a knowledge graph because then it will cause scalability problems. But on the other hand, it has advantages such as data integration is really easy, and it's really easy to integrate several databases. And it also supports verification, and now our data is findable, accessible, interpretable, and reusable within the organization. And also, it's a glass box approach. When I predict between a target and disease, I can have a story to tell that how this prediction works, so it's not really a black box approach. And with that, as bonus, I would like to ask a question to you for knowledge exploration. So the question is, do we really know what we already know? And so this is an interesting question because actually we are reinventing data every day because we don't know what we already know, or we don't know what we already not know. There are so many things happening in different departments or organizations or even different companies. Or like someone in one laboratory, they're doing an experiment which they already checked the interaction between proteins, but other department is not really aware of it. And also these documents they're working on. So if we just link all we know, then we would know what we already know. And knowledge graphs will be really useful for data management and knowledge management in this context. So with that, I thank you for listening me, and I hand over to Mark again. Thank you.
Thank you, Özge. Thank you very much.